and thank you for your attention.
Really, thank you, Professor Chaudhary, for winding up in time, particularly
Okay,
for
good.
maintaining the time.
Yeah, sure.
So, if there is question, we can take one or two questions from the audience. Okay, if there is no question, I have particular one query.
Yeah, sure.
Really, it was a nice talk, and I had that it was quite interesting because I could see that how this active matter is emerging from this microscopic world to the macroscopic world. The thing which you have shared with us.
Several link scales, yes, absolutely.
So I had a particular one query that you had shown one video where you are showing the movement of the proteins.
Yes.
So what is that microscopic technique for capturing such kind of movement?
So let me just first let me go to which one. Is this the one that you're talking about?
Yes. Yes, this one. The next one. Yes.
Yeah. So basically, that's an experimentalist question, and so far as I know, so there are different. So a large number of these, the understanding of these, how these motor proteins move, okay, have happened via what are called in vitro experiments. So what you can do is take these motor proteins and out of the cell. And you can make them active because there is a certain chemical process of doing so. There's an ATP hydrolysis, and these filaments can become active. And again, what you do is attach one end of this filament to of this motor protein. Yes, and these are this is also done to optical tracks. So you have tracks which you have attached one of those filaments, and you see how these filaments are now on. Naturally because of the if you put in. If you have a microtubule or an active filament again on the substrate, it's an opposite experiment to the experiment that I was showing you, the motility assay one. So then what happens is you can actually see how these walks are happening, and you can check whether they are doing a processive motion or they are doing kind of a sliding motion on the surface. Okay. So for kinesin, of course, there are some very detailed beautiful experiments on this in vitro kind of setups. Myosin of showing the sliding motion is slightly more difficult. You can see that there is experiments by what are called by the on-Wales lab. I don't know the details about it, where they actually are also again taken this. Let me. Let me show you the last part.
Yeah, last part. I was interested in the last part. Yes.
Yeah. So sorry again. Let me just stop this, because both of them are starting playing. Okay, so let me just slowly go here.
This one.
So again, what they have done. Yeah. So these are single myosin molecules. Yeah. So they are using AFM techniques to look at it.
Okay. That's.
And you can actually see them. So this how the behavior of it is.
Yes.
Beautiful.
That was really interesting. Yes.
Exactly. It's a fantastic experience.
So I think Mr. Shubhranshu Bose has some query.
Yeah.
Bose, please go ahead.
Hello, Abhishek.
Yes. Follow.
Actually, this molecular motor propagation or dynamics is entropically favorable, entropically or thermodynamically, how it can be explained?
So first of all, one thing, Shubhranshu, as you understand that these are systems which are out of equilibrium, right? There's an activity, there's a chemical energy which is being, and so there's inherently out of equilibrium systems. Okay? Now, what we are, of course, there is so in the molecular model picture, of course, rather I would say if you look at the polymer system, then we can, in the presence of these molecular motors, then you can talk about things like an entropy, which would be the conformational entropy of this, of this kind of extended objects, right?
Okay.
So there are of course thermodynamics of small systems. If you remember, we had discussions on this some time.
Yeah.
So where again, these molecular motors and all these processes all these chemical processes have been studied.
Okay.
Of course, it's a we can talk about that at some point. But yeah, it's so entropy, of course, has a role to play. I mean, entropy is
mainly game of conformational entropy.
If I'm talking about the polymers, yes.
Yes.
Because you can think about how much, if a system is constrained, then the entropy would be reduced. Whereas if it has, if the polymer is in an open environment, then you will have this conformational entropy, which should be much more.
Another form in the Charlies is applicable here. Charlies entropy.
Charlies entropy, I have to think about it. I don't know.
Okay. Okay.
Probably, I mean, yeah, in the molecular motor picture, right? I mean, that's what you're asking. I'll talk to you about that.
Okay, I guess this discussion can be taken beyond the meeting.
Yes.
Yes. Thank you.
Yeah.
Thank you, Professor Chaudhary. Thank you once again for this beautiful talk.
Thank you.
We hope that in future also we'll
absolutely.
Your interaction.
Absolutely.
Thank you.
Thanks.
Thank you. So we have our next speaker, it's Dr. Jhimli Sarkar. So she will be talking about the dissolvable microneedles for better immunization coverage. Let me give a small bio of Dr. Jhimli Sarkar. So Dr. Sarkar is a scientist at Indian Institute of Technology, Kharagpur, and also the co-founder director of Invodigam Private Limited, ID Kharagpur. And she mainly works on the affordable healthcare solution devices. And from the academic point of view, she has obtained PhD degree from Jadavpur University, and she basically works on the nanobio interaction. So, Dr. Sarkar, it's your turn. Dr. Sarkar, I think your microphone is off.
Thank you. Good morning and good afternoon, everyone. First of all, I want to congratulate Shubhranshuda and team to make it happen for their pursuit of creating this interdisciplinary platform. So, let me share my screen. Is it visible? Hello?
No, it's not coming.
It's not coming?
No.
One second. Just. Is it coming now?
Now it's coming.
Yeah.
Please make it full screen.
Yeah, just. Is it okay now?
Yeah, that's fine.
Okay. So, what I'm going to talk about today is something very different from the intricate of science. So, I'm going to share a journey of a product which has been developed in collaboration with ID Kharagpur and my startup, Innovodyne Private Limited. The partial journey, it has not been reached to market yet, but we hope so. So how we have developed this product and how we are dreaming about this product. So I think that will give you all of you, some scenario that can go beyond the intricate of science. So we all know that nowadays vaccine is a buzzword. Wherever we go, there are vaccine. But what is the problem of vaccine distribution? If we can see, this is a real picture. This is a real picture from Puri, a village, a remote village from India, where these two women, braving the harsh weather, trying to distribute vaccine in the remotest corner of the world. But this is a real problem in vaccine distribution nowadays. According to who has implemented various action plans like 2020 Global Vaccine Action Plans, strategic goals for 2030. In spite of all these plans, up to 50% of vaccines are wasted globally every year. And as I have given the statistics, how much coverage has been dropped from earlier year to 2020. And this is very, very astonishing or very fearing subject that 23 million children under the age of 1 year did not receive basic vaccination. And it is very alarming nowadays. And in case of COVID-19 also, what we have seen that only 10.6% of people in low and middle income countries have got the vaccination. So this is a real world problem. And the problem lies in this scenario. Like mainly the cold chain facilities are not available in the remotest corner of the world. And also there are health worker unavailability. Every time we cannot have access to the health worker who can give the vaccines to the needy. So 54% of the delivery cost and 6% of the supply chain cost of the total vaccine cost comes from this problem. And another major problem is the generation of sharp waste and vaccine loss. As we all know, that there is a high chance of cross-contamination while giving vaccine through needles, which is a conventional way to give vaccine. Now 33,000 HIV infection, 1.7 million hepatitis B and almost over 3 lakh hepatitis C infections occur every year due to these unsafe injection practices. So what is the solution? Here comes our solution. What we propose a thermostable vaccine patch instead of vial and needle. Imagine you have a patch. You apply it on your skin, press it for 30 seconds, remove it, and voila, your vaccination is done. So what are the values it adds? It doesn't need any cold chain. So your cost is reduced and availability of vaccine to the remotest corner increase. Next, there is no vaccine loss. Because when you open a pentavalent vaccine vial, almost 80%, 60% in average vaccine is being lost because of certain conditions, like maybe people are not coming on the proper time, health worker not capable to administer vaccine. These all are the problems which results in vaccine loss. Next, it can be self-administered. Suppose in a pandemic situation, you have this patch, you need not to go out from your home. You just have this patch delivered at your doorstep and you can apply it. Next, there is no sharp waste generation and less cross-contamination. And finally, this vaccine patch, while applying, you cannot receive any pain. Why? I'm going to discuss in details. So how it works? Actually, this vaccine, this is a dissolvable microneedle patch. When it penetrates the skin after pressing, it comes into the contact of the tissue fluids and the polymer, which are of course FDA approved. gets dissolved within 10 minutes after application. And it doesn't go up to the layer where nerve cells are present beneath the skin. So the pain cannot be felt. And this is the way the vaccine works. Now, why this is more efficient in delivering or in producing antibody? Because under the skin, there are more antigen presenting cells than intramuscular injection. So we expect that in smaller dose, the vaccine can evoke more immunogenicity. So this is the vaccine patch we have developed in our lab, and this is the microneedle, which is the same picture. You can see the structure of the microneedle. It is a pyramidal structure, as you can see. So it is pyramidal because we have done simulations and various structural study, and we have seen this kind of structure penetrates very well inside the skin without breakage. Now, this is the intricate of the science, that how we are doing this. We are using sorbitol polymer, which are FDA ICMR approved. All the things all the ingredients we are putting here are FDA ICMR approved. Now, this is the main procedure of how we are molding this polymer into a vaccine patch. Specifically, we are using micromolding techniques and then removing these micromolding techniques from the mold. We are using various kind of mold. First, master structure we create. Then from there, we create a PDMS micromolding structure. Then from there, the third step, we create the sucrose backing solution. And finally, we demold those things, dry it up, and package it so that we can get the final product in aseptic condition, of course. This is some of our study in vitro and in vivo, where we have studied that the vaccine is really stable at 40 degrees centigrade up to 90 days in vitro and in vivo. We have studied in vitro in the we have measured the IL-2 concentration in vitro, and in vivo we have studied this through ELISA technique. And this is some of our results where we have done penetration assay in rat skin and mice skin, and we have calculated the force which we require to penetrate the needle inside the skin, and we have found that is around 0.26 Newton for compressive force, which is sufficient to press the vaccine inside the skin without the breakage of microneedle. And what value is going to add? The first and foremost necessity, that is the coverage. It has been predicted that if the original 5-in-1 pentavalent vaccine was reformulated as a fully thermostable preparation, its availability will go up to 100%. So that is the main value we are going to add in the chain. And it is also a risk-free vaccine administration procedure where no cross-contamination is possible, and also, as I have mentioned, it is painless and self-administrable. We have also done some market study. Why it is necessary? Because we don't know whether this product can be brought into the market. So what we have seen that at around 2028, the global vaccine delivery market devices will grow up to 6.85 million. And this proves that, and India holds the 44% global capacity in vaccine production. So this proves that this kind of devices has huge market in future and huge demand in future. Vaccine is the buzzword, because when you are in home and the vaccine is being delivered to you at your doorstep, then it is very, like, we need not to queue in the clinics. We just have the prescription and we can have the vaccine. So it is acceptable to the audience and customer and consumers also. So this is our 2 years plan. We are now technology ready level 3, where proof of concept has been developed and in vivo assay has been done. And now at the end of the 12 months, from Innovodyne Private Limited, we are going to make it to the next level, which is technology ready level 4, and within 24 months we hope to file the DCGI application to get the approval of the vaccine. These all are the grants we have received so far, and we have been supported by BIRAC big grant, which is a grant from Indian government to commercialize the product for the startups, and ICMR, Indian Council of Medical Research grant also. So thank you. Any question?
It was really a nice talk from Dr. Sarkar, and we have learned about the product of the vaccine patch, and which can be a solution for the conventional vaccination system. So the talk is open for questions. If there is any query, please join. I think Professor Abhishek Chaudhary has.
I had a question. Yeah, very nice product. It's good to see this. One question is that these microneedles that you talked about, are they going to be like, are they supposed to dissolve inside the skin, or
Yes.
just,
Yes, they are supposed to dissolve inside the skin. It's dissolvable microneedle.
Okay, I see. The other thing is that with such kind of, I mean, are there such vaccine patches available elsewhere in the world, or is it like
No, right now, university in USA, they are also trying to develop this kind of patches. There are different kind of vaccine patches, not vaccine patches, microneedle patches are like in clinical trial. They, one of them have crossed the second phase clinical trial also, but no vaccine patches are available right now in the market.
Right.
So we are the first in India who are going to bring hopes
Yeah,
so
very nice.
this patch in the market. So I don't know, but we'll try our best.
Sure. It looks like a very promising thing to do.
Thank you.
Thank you.
Dr. Shobhan Bose has some query. Please go ahead.
Yeah, Professor Sarkar, first of all, thank you for this nice talk. Though I missed a little bit. And my point is that you told already in response to Chaudhary that microneedle will be dissolved, right?
Yes.
In the body. Now, any part of this patch will be thrown away.
Yes, that is the backing part, which is made up of sucrose, only sucrose. That is the backing part.
Whether from there any kind of contamination is possible.
No, it's only the backing sucrose part. It doesn't contain any vaccine. So it's the sucrose material which can be like biodegradable. There is no contamination.
There is no contamination. Okay. So that kind of things already you tasted, right?
Yes.
Okay, that's a great job in that case. Really
Thank
great.
you very much.
Thank you.
Okay. Is there any other query?
I have a query.
Yeah, please go ahead.
Jimniti, you have told that some portion of sucrose is not dissolved. So, is there any possible that it can be consumed by our body?
No. The thing is that there are two parts. One is the microneedle part, which is of different material, different polymer, and the backing part is the sucrose part, okay, which is being peeled off after just like band-aid. Consider it is a band-aid. Okay, you press the band-aid and you pull it out. The backing part is being pulled out. So it's like that. The pulled-out part is the sucrose part, which doesn't contain any vaccine. Only the middle part, which is getting penetrated, contains the vaccine and which is completely dissolved under the skin. So there is no contamination probability. I'm able to give you the answer.
And you have told that anyone can take it home easily. So it is that much of friendly, it can be.
Yes, but you know, when the product is being developed, there are certain regulatory things which you have to cross, the hurdle you have to cross. It can only be done if the government gives us some push-pull interaction. Like if government or regulation allows the doctor that you can prescribe it and you can, one can purchase some vaccine and administer it on their skin, so it's okay. But the feature is there. But I don't know whether government will permit us for self-administered things or that I don't know. But the feature is there. It can be pressed. You can on your own press on your skin and press it for 30 seconds and it will be done. So we have had that value.